Okay, so for my video, I'm going to be doing mitosis and meiosis comparison. Before that, let's learn a little bit about mitosis. So mitosis has 5 phases: interphase, prophase, metaphase, anaphase, and telophase. Interphase is basically when the cell, when the chromatin inside the nuclear membrane are starting to replicate. And as you can see over here, the centrosomes are still kind of away, they haven't split apart. In prophase, the DNA starts replicating itself and the chromatin actually go on each side and they start pulling the nuclear membrane apart. Metaphase, you have a full lineup of all the chromosomes with the 2 centrosomes on each side, kind of pulling it with microtubules. Then you have anaphase, where that cell actually starts pulling itself apart. As you can see, there's a break right there down the middle. The chromosome is starting to break. And in telophase, we have what's called a cleavage. We have the beginning of 2 new daughter cells that are genetically identical to the parent cell. Right here, we see the cleavage, and eventually after this, cytokinesis will happen, in which the 2 cells will split apart, making 2 new identical cells, in which, if need be, they can go ahead and continue with interphase and start the process all over again. Okay, so in review, mitosis, for example, if we're looking at this, each mitosis cell has, if we're speaking about humans, say it has 46 chromosomes. They then divide into 2, forming 2 daughter cells. And this 2N signifies the 46 chromosomes. So they have the same number of diploid cells. Now these are called somatic cells. They make up most of the human body. They can be found in liver, skin cells. So they're the most plentiful cells we have in the body. With meiosis, it pretty much goes through the same exact phases that mitosis does. It goes through the interphase, prophase, metaphase, anaphase, but then it just goes a little step further. So with meiosis, we're primarily dealing in fertilization. So we're looking at, for example, in human sperm cells and egg cells. So with meiosis, again, we start with that same diploid cell like we did in mitosis, and we start splitting the homologous pairs. So we end up getting 2 haploid cells. So remember from the first one, mitosis, this had the 2N signified 46. Now the 23 signified 23 chromosomes. So that's so actually divided in half. Now remember, mitosis, these are the same exact daughter cells, or exactly like the parent cells. With meiosis, it's different. They're not identical to their parent. So these 2 23 chromosome cells, they then further break apart and create 2 of themselves to form gametes, or sex cells. So in the case of, let's say, sperm, the sperm cell created 2 more of itself, and that'd be called meiosis 2. So meiosis 1 is when you take the diploid cell, you make 2 haploid cells. Meiosis 2 would be when you take that haploid cell and then you make 2 more. Okay, and again, they're called gametes. And meiosis, these cells are different. They're called germ cells. They're a little different than somatic cells. These are just for replication, so reproduction. I'm sorry, not replication, reproduction, excuse me. And these are, again, somatic cells. Key differences between meiosis and mitosis is just for it's complete replication. So of a cell, think liver, think skin, think anything that needs to heal and needs to make more of themselves identical. Meiosis is solely for reproduction. So if you're thinking of plants, you might think of trees producing more pollen to spread out more of themselves. If you think of humans, you're thinking of sperm or eggs.